Hello, students. So today we will discuss antibodies, their structure, their function, and their types. So in human antibody isotypes, so basically these isotypes, we see that there are these different isotypes, and these isotypes are based on the heavy chain. There are 5 heavy chains, 5 different types of heavy chains. And with reference to this, or relating to this, the name of antibody is, for example, IgA, IgD, IgE, IgA, IgM, IgG. So in IgA, this isotype is alpha chain. Heavy chain is of alpha type, and D is IgD is delta chain. Type of heavy chain is delta chain, and IgE is epsilon, and in IgG it's gamma, and in IgM it's mu. So when the heavy chain is alpha, then the type is IgA, and when the heavy chain is delta, then it's IgD, and epsilon, then IgE, and gamma, then IgG, and mu, then IgM. So there are on the basis of these isotypes, there are 5 different types of isotypes, and we have 5 different types of antibodies. So isotype of antibody, that if it is IgA, it means it will have alpha chain. And subtypes are alpha 1 and alpha 2, IgA 1 and IG2. So there are 2 types of alpha chains, IgA 1, IgA 2. 95% amino acid sequence is similar, both of these. There is a small difference in the amino acid sequence. That's why they are termed as IgA1 and IgA2, or alpha 1 and alpha 2 isotypes. Serum concentration is about this 3.5 milligrams per ml, and serum half-life is 6 days, and they are secreted in the form of this. You can see that there are 2 antibodies linked with the help of secret this J chain or joining chain, one antibody and the other antibody. So it's dimeric antibody, and it will, most of the IgA will be secreted, and daily our body will secrete about 15 grams of IgA. So it will help in the first line of defense, or it will help components of innate immunity or mucosal immunity, where it will help and fight with the microbes and bind with the microbes so that they will not be able to enter in the body. So the first type is IgA, and the second one is IgD, and these subtypes, there are no subtypes, none, and there's only one type of chain, delta chain, and its amount is very less, and its half-life is 3 days, and this secreted form, it is not secreted, and it is only present on the naive B cells, antigen receptors. So it has no not so important function in the body. So it's not so important. Then we see IgE, it has no subtypes, and the heavy chain is of type epsilon, and this amount is 0.05 milligrams per ml, and its half-life is 2 days. And here you can see that this is the IgE. In IgE, these are the FAB regions, and this is the FC part. And FC part, you can see that it have this epsilon chain, constant CE1, CE2, CE3, CE4. So it helps in defense against helminthic parasites, and it is involved in the allergies and type 1 hypersensitivity. So type 1 hypersensitivity response will take place with the help of IgE, and here it is not working to aid in the immunity, but it is a disadvantage of the immune system, because it will result in allergic response, and we know allergies are not good. IgG, so there are 4 subtypes, that is IgG 1, 2, 3, 4, because there are 4 types of gamma chains, gamma 1, gamma 2, gamma 3, gamma 4, and 95% amino acid sequence is similar. Just 2 or 3 amino acid sequence will be different, but it will make it different, and it will change its abilities. So the amount is 13.5. So the about 80% of the antibodies in the blood sera belong to the IgG. And the half-life is about 23 days. And here you can see this IgG1, and these IgG antibodies are in the monomeric form, IgE in the monomeric form. IgD is never secreted, but if it is, it should be in the monomeric form. So in IgG, we see that it helps in opsonization, it will help in the activation of complement system, it will help in the antibody dependent cell-mediated cytotoxicity, and it will help in neonatal immunity and feedback inhibition of B cells. So the best antibody for complement activation will be IgG3. IgM, no subtypes, and heavy chain is mu, and the amount is 1.5 milligrams per ml in the blood in the serum, and half-life is 5 days. And it is, when it is present on the B cell, it is in the monomeric form. But when it will be secreted, then it will be in the pentameric form. You can see that these 5 antibodies, 1, 2, 3, 4, 5 antibodies will come together, and they will form one antibody, that is IgM. It's the strongest antibody and have 10 binding sites, and it show more affinity and more avidity. So basically naive B cells will produce this, but when it will be secreted, then it will be in the pentameric form, and it can help in the complement activation. So you can see that there are 5 different types of antibodies. It depends on the heavy chain they have. If it have alpha chain, then it is IgA. If it have delta chain, then IgD. It have epsilon, then IgE, and gamma chain, then IgG, and mu chain, IgM. The first antibody in any immune response which will produce is IgM. And the antibody which is secreted mostly is IgA, and a small amount of IgM also. And the mother's milk will have IgG in it, so which will help this newborn child to support its immunity and defense. Okay, thank you.